Hi everybody. This will be my audio lecture for chapter 18, immune disorders. So what this chapter is going to be talking about are a number of different diseases and disorders that are the result of some issue with the immune system. So either the immune system is overacting, not acting at all, acting when it's not supposed to, things like that. So everything that you know about how the immune system is working, something is not happening appropriately in each of these different disorders. So the first group of disorders they're going to talk about are hypersensitivities. And so these are basically like an overreaction of your immune system, or like an immune response that is happening kind of beyond what would be reasonable. And there's 4 different types. There's type 1, which is also known as immediate, type 2 or cytotoxic, type 3, immune complex mediated, and then type 4, which is delayed or also known as cell mediated. So I'm going to talk about each of these. So type 1, an immediate hypersensitivity, these are basically your allergies. So this can be a localized immune reaction or a more systemic immune reaction. The result is a release of inflammatory molecules in response to a particular antigen. And in this case, we're talking about antigens known as allergens. So if you have allergies, or if you know people who do have allergies, those would be things like pollen, pet dander, other types of environmental allergens, food allergens, things like that. And these are called immediate hypersensitivities because the reaction is pretty quick upon exposure to those antigens or allergens, so within seconds or minutes. These types of hypersensitivity, so allergies, they happen in like a 2-step process. And those of you who have seasonal allergies, or if you have food allergies, you might be more familiar with this because you don't, I know it's true for me, I have, very mild seasonal allergies, but I never used to. And I know people who have food allergies that didn't always have food allergies, and that's because of this 2-step process. So when you're first exposed to a particular allergen, you are sensitized to that allergen, and I'll show you what I mean in a second. You may not have an actual physical response yet. There's stuff going on in your cells, but you don't have the release of the inflammatory chemicals yet, and it won't be until maybe the second time you're exposed, or the third or the fourth time you're exposed to that allergen. And that's when what happens is those sensitized cells degranulate and release all these inflammatory chemicals. So let's look at the picture because I think it makes a little more sense. So this is showing you the sensitization step that happens the first time you're exposed to an allergen. So here's our allergen up here. There's an antigen presenting cell that encounters that allergen. There's foreign antigen on the surface. Your body recognizes it as foreign. The antigen presenting cell takes it to a lymph node, presents it to a helper T cell, and in this case we're involving B cells. So this is kind of sort of the antibody mediated response. So what has happened? We've activated a B cell because of this antigen. Plasma cells develop, release antibodies designed for this allergen or the antigen, and instead of going and finding more antigen, the antibodies go out into the blood and find your granular white blood cells. Granular meaning these white blood cells have a whole bunch of granules inside of them, which are the inflammatory chemicals. And so the sensitization part of this is when the antibodies that were generated in response to the first exposure to this allergen, which is the antigen, the antibodies go and find these particular white blood cells and latch on to them. And remember that the antibodies are designed to bind to the allergen. So now we have these sensitized cells. Nothing physically, you don't really feel anything physically in this step. It's when you're exposed again. And that's the degranulation. So what happens when you are exposed again to that same allergen, those are going to bind, those allergens will bind to anything that it can bind to. And in this case, it's going to be the antibodies that were made the first time around. And those antibodies tend to be stuck now on these granulocytes. And when the allergen attaches to that antibody, it tells this white blood cell to release all of its inflammatory chemicals, not anti-inflammatory, inflammatory chemicals. So like histamine, leukotrienes, other things, all those things that are going to cause immune responses or immune symptoms. So like runny nose, watery eyes, itchy watery eyes, itchy throat. Think of like that in terms of seasonal allergies, or like a rash or something like that, like hives, or even a little more serious like anaphylactic shock, where you have major edema, swelling of your respiratory passages, and now you can't breathe. So the severity of the effects really depend on how many of these sensitized cells degranulate at the same time. So let's see. They're talking about the specific roles of the different types of white blood cells that can degranulate. So like mast cells, basophils. In this table, they're talking about the different types of inflammatory chemicals that can get released and what do they do. So again, depending on which of those cells degranulate and how many, and therefore that will determine what kinds and how much of each of these inflammatory molecules are released. That will determine the severity and the kind of the location of the symptoms. Is it a respiratory, like is it just in your eyes and your nose, or now is it in your air passages? You don't have to know the roles of these different white blood cells, but that they're just giving you the information so you have a little more information about those cells. So normally those signs and symptoms can be mild. So like hay fever, which would be seasonal allergies. You know, it's just kind of annoying. Even seasonal allergies can vary in severity. You know, for me, it's not really a huge deal. It's mostly like a constant drippy nose. But for other people, it's a lot more debilitating, and hence that's where different types of allergy medicines and shots and things like that might be more important. When we're talking about anaphylaxis or anaphylactic shock, that is when a lot of those mast cells degranulate at once. Lots of histamine is released at once, and it overloads your body and causes major swelling of your airways. And that is a life and death critical situation. And you need to treat that with a very quick and powerful vasodilator like epinephrine. So bee stings can cause this. Certain foods, peanut allergies, other allergies that people have to different foods can cause this severe reaction, and those are the people that have to have an EpiPen with them. So EpiPen is short for epinephrine, and you need that, otherwise it's a life and death situation. So this is showing you if you have allergies and you've had these tests done. I have not, but I know people who have. This is a way to test to see what exactly you're allergic to, where they can inject just a tiny bit of the particular allergen under your skin and look for basically a skin reaction. So it's like an itchy spot, and they can see the severity of the reaction, and then the doctors can design a course of treatment for you. Sometimes you can grow out of allergies, you can grow into allergies. Allergy shots are one way of kind of helping you grow out of them in a way. They're kind of weird, and anything that's dealing with the immune system is in general just a little bit weird, and in some respects a little unpredictable, because there's a lot of different things that can kind of trigger or change your immune system and make it more sensitive or less sensitive. So allergies are kind of one of those things. So that's type 1. Type 2 hypersensitivities are known as cytotoxic hypersensitivities because cells are destroyed, cyto and toxic, by the immune response. So your immune response is actually causing cell death. Usually when we're, in terms of examples, we're really talking about blood typing and blood types that do not go together and are incompatible. So let me show you the picture while I talk about this. So if you took ANP2, you already know this. If you're in it right now and have just started or haven't taken it yet, you might not be there just yet. But in a nutshell, your red blood cells have antigens, self-antigens basically, on their surface, and that there are different combinations of antigens that could be on that surface. So in this example, these are the host or the patient's red blood cells in this blood vessel right here. The ones with the circular antigens on their surface. Those antigens are type A antigens. So this patient has type A blood because his blood cells, red blood cells, have A antigen on their surface. Those people with type A blood also have in their blood anti-B antibody. So this antibody is designed to bind to B antigen. So this person doesn't have B antigen on their red blood cells. They have A antigen. You don't want antibodies that will bind to your antigens, because if you do have that, then you're going to get clumping of your red blood cells. You don't want that. So this is a type A person. He has type A blood, meaning A antigen on the surface of his red blood cells, type B or anti-B antibody. In this situation, this patient had a blood transfusion, but the blood is type B blood. This is the donor blood. So this is blood, red blood cells that have a different antigen, type B antigen, on the surface of those red blood cells. Your body's going to see that as foreign. When these red blood cells mix in with the patient's blood, his anti-B antibodies are going to bind to those B antigens on the donor blood and cause clumping or agglutination and trigger the formation and the release of complement, which is going to lead to cell lysis. That is one of the results of complement activation, if you remember from chapter 15. So this causes lysis of red blood cells. These are the donor red blood cells, but that can lead to problems in general in the patient. But also, if the patient needs blood and that donor blood was just destroyed, well, the patient is still in need of blood and there's going to be a problem. So this incompatibility of blood type is leading to antibody action that causes cell death. And that's where the term cytotoxic hypersensitivity comes into play here. Another example is this hemolytic disease of the newborn. So this has to do with a different antigen that may or may not be on the surface of your red blood cells, which is this Rh antigen. So if you have a positive in your blood type, that means not only do you have either A or B or none or both of those antigens, but you also have the Rh antigen on your red blood cells. So the problem here is if a woman is Rh negative, and I'm going to show you the picture here because I think it makes more sense. So in this case, the mother is Rh negative. That means her red blood cells do not have the Rh antigen on their surface. And in her first pregnancy, she has a fetus that is Rh positive. So the baby's red blood cells do have Rh antigen on their surface. So during delivery of this baby, there could be some breaks in the placenta, and some of those baby's red blood cells mix with the mother's. And so the mother's blood is now encountering these different red blood cells that have this Rh factor on their surface that is foreign to the mother. And so the mother's immune system will develop antibodies against that Rh factor. Now this baby is not harmed in any way because the baby was already born. But now the mother has these anti-Rh antibodies, which she did not have before. So the problem is in a subsequent pregnancy, if that second baby is Rh positive, now the mother's antibodies are going to attack that baby. And it's going to basically cause a miscarriage. And so for women who are Rh negative, even during their first pregnancy, they're given a drug called Rogam, which prevents that mother from forming these antibodies. And every time she's pregnant, despite what the baby is, because you don't want to wait to find out what the blood type of the baby is, as soon as that mother is pregnant, again, at any time, she'll immediately start taking that Rogam to eliminate the possibility of forming any type of antibodies against that possible baby. All right, so those are type 2. So cytotoxic, we have antibodies that are being activated and causing cell death. Type 3 hypersensitivities are called immune complex mediated hypersensitivities because there are these immune complexes that are left over from a normal immune response that are not cleaned up afterwards, and they're causing problems. So we have another picture here which I think is good. So we've got antibodies and antigens during some normal antibody-mediated immune response, some kind of an infection. Okay, you created antibodies against the antigen. The antibodies are binding to the antigen, and they're doing what they're supposed to do. And normally, at the end of the infection, we need to clean up these complexes, these antibody-antigen complexes. And that's the job of some phagocytes. Let's get rid of these antibodies that are bound to these foreign antigens. We need to get rid of this stuff. Sometimes some of these complexes go unnoticed by those phagocytes, and they kind of stick to your blood vessel walls. The infection is over, but we have some leftover antibody-antigen complexes. And this can activate complement at a later time, kind of for no reason, really. There's no infection, but antibodies, one of their jobs is to activate complement. And so if the antibodies are there and they're active, even if there's no current infection, they're going to activate complement when those proteins come by. So they are activating complement, and what kinds of things can complement do? Go back to chapter 15. But they can attract neutrophils, and they're going to release enzymes. That's part of the innate immune response. They can release other inflammatory chemicals, and so it's kind of misguided. It's a misguided immune response. It's an immune response in response to nothing. There's no infection, But these inflammatory chemicals are released, hanging around, and they're going to cause tissue damage. And so this is the case of, rheumatoid arthritis. This is a type of type 3 hypersensitivity, and as we'll see later, it's also an autoimmune disease. So it falls under both categories, but what's happening is there are antibodies kind of that are being made against your own antigens, which you don't want to happen normally, and that's how we call that autoimmune. And these complexes are getting formed and hanging around and causing tissue damage due to the kind of chronic release of these inflammatory chemicals. And it happens to occur when you're talking about rheumatoid arthritis. It's occurring in your joints. And so you have constant release of inflammatory chemicals. It's going to cause tissue damage, and when you talk about joints, this is what's going to happen when it's happening over a long period of time. So that's very debilitating, and it can lead to other complications, which can lead to death if it can't be controlled and managed well. Lupus is another type of type 3 hypersensitivity, and it's also autoimmune. So autoimmune meaning that you've got antibodies against your own antigens instead of foreign antigens. And then in this case, it's type 3 hypersensitivity because it's causing these immune complexes that are leading to tissue damage. All right, so then the last hypersensitivity is type 4, which is delayed or cell-mediated. So think of things like poison ivy, poison oak, those types of skin rashes because you've come into contact with some oil, and it's kind of like an allergy, but it's not immediate. You might rub up against poison ivy and not realize you've done that until the next day, and that's when you see the rash. So it's an overactive immune response to a particular antigen, but because the effects aren't seen immediately, we call this a delayed response, or type 4. Another example is the tuberculosis response. So if you've ever gotten a TB test, they give you the little injection under your skin, and what are they doing? They're giving you antigens of tuberculosis, of that bacteria under your skin. And then you have to come back a week later, and they're going to look at your arm to see if there's a bump there. Well, if there's a bump, it means that you have tuberculosis antibodies in your body that have reacted with the tuberculosis antigen that was injected under your skin and is causing a very localized immune response or immune reaction. What would be reasons for having tuberculosis antibodies in your blood? One, if you're infected with tuberculosis, so a positive TB test could suggest that you actually have tuberculosis, or, and this isn't always true for us in the US, it might be if you work in the healthcare field, but if you have been immunized or vaccinated against tuberculosis, you will have tuberculosis antibodies and you will have a positive TB test. Now, I knew somebody who was born in India, and before he moved here, he had to have a tuberculosis vaccine, and he worked in the healthcare field, and he had to get a TB test every year, and every year it always came up positive. And he's like, no, it's because I had the vaccine, and so it's causing some issues. But that's what that's another example of this delayed hypersensitivity. So this is kind of what a positive TB test could look like. This just means you've got swelling, you've got redness. Those are all very innate immune responses, but it's an inflammatory response as that can be a result of the reaction between antibodies and antigen. And then the others I had mentioned earlier, contact dermatitis. So that's poison ivy, poison oak. If you have a latex allergy or an allergy to formaldehyde, those kinds of things. the reaction might not be immediate, and so then it falls under this type 4 category versus type 1. Another type of delayed or type 4 hypersensitivity would be tissue transplant rejections. That's not immediate, but you're basically, if you don't take anti-rejection drugs and you get somebody else's kidney, those cells have their own antigens on their surface. Think of their own MHC antigens on their surface from that person. Your body sees those antigens as foreign, and it's going to mount an immune response against that entire organ. It won't be immediate, but it will happen, and that's why you need rejection drugs. They also talk about here different types of tissue grafts or transplants. So let me kind of explain. So an autograft would be like a skin graft. So let's say you had a very bad burn somewhere in your body, and they need to do a skin graft. They can take skin from another part of your own body and replace the burned skin. Since that tissue and those cells are coming from you, that's auto. So there's no issue of rejection here. An isograft is a tissue graft or a tissue transplant between two different individuals, but they're genetically identical. So like between identical twins. There might be a little bit of risk of rejection here, but not as much as the type of tissue transplant that we are most familiar with, which is allograft. So that's like you get somebody else's kidney, you get somebody else's heart. Two different people, genetically different, but of the same species. And then the other type of graft is a xenograft. So now we're talking about two different individuals, one donating tissue to the other, but the two different individuals are different species. Sometimes that can happen. There have been successful I think like pig heart transplants, baboon heart transplants, other tissue transplants in humans using organs from different animals. It's not very common, and there's a lot more risk of rejection, but it can work if absolutely necessary. All right, so here's a nice summary of those 4 different types of hypersensitivity reactions. It's nice that it gives you the causes here, the time course, meaning how long does it take for the reaction to occur, and what kinds of cells of the immune system are involved. You don't necessarily have to know those details for the test, but that is there if that helps you understand it better. All right, so the next part of this chapter talks about autoimmune diseases. So autoimmune means your immune system, instead of attacking foreign antigen like it's designed to do, it's attacking self-antigens, meaning it's attacking your own cells, your own tissues, your own organs. Why would your immune system do this? There are a lot of reasons. I'm not going to go through all these reasons, and I don't even think the ones listed here are all of them, but this is really a testament to how, I would say, finicky your immune system and sensitive your immune system can be. I mean, even think about like when you're stressed, do you tend to get sick? Some people do. I mean, stress can play a role on how well your immune system works. So in that sense, I'm not saying stress can cause autoimmune disease, but it's just meant to illustrate how sensitive your immune system can be. So there's lots of different reasons that an autoimmune system could be triggered in a person. Often these are not diseases that you're born with. You end up kind of acquiring them through your lifetime. I've known some people who've acquired an autoimmune disease after having a surgery. So like the actual surgery put stress on your immune system because it requires tissue repair and things like that. And that stress on the immune system kind of put things out of whack with the immune system itself. And now that person has an autoimmune disease. So lots of different reasons. Just keep these in mind that are on this list. So there are two major categories: systemic autoimmune diseases, meaning the immune system is attacking a variety of different cells all over the body, or more localized autoimmune diseases where the cells that are under attack are more localized to a particular organ. And so there are lots of different autoimmune diseases out there that can attack any variety of cells that we have. And then what they do in the rest of these slides is kind of just go through a few of the different diseases. So let me just talk about some of them. You can read about the other ones. Type 1 diabetes, that's autoimmune. So that's affecting your pancreas, particularly these islets in the pancreas, which are endocrine cells. So this would be an endocrine autoimmune disease. And so you've got an immune response against your pancreatic cells. That's going to affect how well your pancreas can regulate your blood sugar. So that obviously is going to be a problem. Graves' disease is an autoimmune response against your thyroid gland. What it does is causes overactive thyroid, and that can lead to a goiter, which is it looks like a big swelling in your neck. Multiple sclerosis. This is a degenerative disease. This is when your immune system attacks the myelin sheaths that cover the axons of your neurons. And if you go back to ANP1 and think about what myelin is doing, that's an insulator basically, and it's allowing nerve signals to be sent down those axons very quickly. If an axon is designed to have myelin and does not, then the nerve signal travels so slowly down that axon that it's like it basically doesn't travel at all. And so if you can't send nerve signals, think of that, if that axon is trying to talk to a muscle, now you're not going to be able to use your muscle. So, and again, this is due to your immune system attacking those myelin sheaths. Rheumatoid arthritis, I talked about earlier. The thing to think about with autoimmune diseases is how do you treat that? And the only way to do that is through immunosuppressive drugs. And so if you've seen, I'm sure you've seen on TV, they advertise drugs on TV all the time, unless you're exclusively watching Netflix, which I know some of you do. But if you watch live TV and you see commercials, you'll see, I can't remember the name of the drug, but it's the same drug designed and advertised to treat RA, which is rheumatoid arthritis, and then also psoriasis, which is an autoimmune disease of the skin. It's the same drug. So how can one drug treat these two seemingly very different diseases? And the answer is they're not really that different. They're due to autoimmune function or autoimmune disease function. And so the drug is really just a general immunosuppressive drug. The problem with that is, yes, you can control the autoimmune disease, but you're suppressing your immune system. So that puts you at risk for other infections throughout the rest of your life, basically. So, it's something that a patient really has to kind of think about. How debilitating is the disease? Can I suppress it just enough to live comfortably and not affect my overall immune function? All right, so we talked about hypersensitivity. So that's overreaction of your immune system. There were 4 different types. Then we talked about autoimmune diseases. So that's when your immune system is attacking your own cells instead of just foreign cells. And now we're going to talk about immunodeficiency diseases. And the bulk of this section really is talking about HIV and AIDS, but there are a few other things to mention. So we're talking about immunodeficiency diseases. That means your immune system is not working. It's not attacking your own cells. It's not overreacting. It is not reacting enough at all, and so your body can't fight infection. There are two types. Primary immunodeficiency diseases. These are things that maybe you're born with or you develop early in life. And they talk about a few examples, but I'm not going to go into detail about those, and you don't really have to know any of those. The big one is the acquired immunodeficiency diseases, which you develop later in life. Usually there's some cause to it. There's a reason for that immunodeficiency disease. And honestly, the only example they give you in the book is HIV and AIDS. So we're going to spend most of our time talking about that. Here is a list of some of those primary immunodeficiency diseases. They give you the name. They tell you what the problem is in your immune system. What are you lacking? And then what happens as a result. And that's basically the bulk of what you have to know. We're going to spend most of our time on HIV and AIDS. So, excuse me. Acquired immunodeficiency syndrome. That is AIDS. This is the immunodeficiency disease. And I mentioned earlier that these immunodeficiency diseases often they have a distinct cause. In the case of AIDS, the cause of AIDS is a virus known as human immunodeficiency virus. You cannot get AIDS without having been infected with HIV first. Okay? So AIDS stands for what I said before, acquired immunodeficiency syndrome. So it's not just a disease, but it's a syndrome. And so what do we mean by that? Meaning it's something that does have a cause to it, but there are other characterizations or characteristics that have to accompany infection with HIV. So what does it mean to have AIDS? It means that you need to have an HIV infection, so you have to have HIV or human immunodeficiency virus in you. You also have a low number of helper T cells, and I'll talk about why in a second. And then you also have a collection, a variety of certain opportunistic or rare infections. So the combination of all three of these things is what tells a doctor that a patient has AIDS, and also why we call it a syndrome and not just a disease, because you can have a variety of other infections as the result of having an HIV infection to begin with. So you need several infections and diseases, positive tests for HIV, and low helper T cell count for you to be qualified as having full-blown AIDS. All right, so what are the diseases that often happen that are associated with AIDS because of the HIV infection? I'll talk about why in a second. They're giving you a list of different diseases here. You don't have to know what causes them necessarily. You don't have to know what organs they target. Just have a vague understanding that, yes, there's going to be a lot of different types of infections that you're going to get more frequently after having been infected with HIV and if it goes untreated. All right, so let's talk about HIV. So human immunodeficiency virus. This is the pathogen that will lead to AIDS if infection of HIV is not controlled. So the virus itself is an envelope virus. It's a positive single-stranded RNA virus, but it's also a retrovirus, which means it will use reverse transcriptase and do transcription in reverse to make double-stranded DNA from its single-stranded RNA. The advantage here is that our cells have double-stranded DNA, so if this virus can make double-stranded DNA, then that DNA can get incorporated into your chromosomes and become part of you permanently. And I posted a couple of web links in eCampus. If you look under helpful videos, under the HIV videos, a couple of different web links that talk a little bit more about the replication cycle of HIV and illustrates how that can happen. Two types of HIV. The one that I think is more common, especially in the United States, is HIV-1. And that's what we're going to talk about here. So let's look at this structure. I'm going to show you a picture. It is an enveloped virus. So here is the envelope. Here is the capsid. Here is the single-stranded RNA. And there are two important proteins that you find on the envelope, GP120 and GP41. GP120 is involved in the attachment of the virus to the target cell. It is also the, I guess we consider this to be the antigenic determinant of this virus, meaning this is kind of serving as the antigen and what your body is responding to, what your immune system would respond to try to fight this. But it also mutates frequently and can change a lot, which is why we don't have a vaccine against HIV yet. GP41 is involved in the uncoding of this virus. So I'll show you a picture in a minute. So here we go, replication cycle of HIV. So this is a virus, so remember, it has to attach to the host cell, that the genome, in this case, the single-stranded RNA has to enter the host cell, and then it's going to undergo synthesis, meaning it's going to make more copies of viral genome, more copies of viral protein, assemble them, and release. And what's interesting here is release and assembly are reversed in HIV. So when the virus actually is released from the cell, it hasn't been put together yet, and it gets put together after it gets released. I'll talk about that in a minute. So we have attachment here through GP120. I'm going to show you another picture that shows this attachment and entry process in a little more detail in a minute. So we have GP120 attaching to in this case, the receptor that HIV will attach to is called CD4. And if you recall, we usually associate CD4 with helper T cells. There are some other immune cells that also have CD4 receptors on their surface and therefore can also serve as a target for HIV. But the key thing is, all right, we have this virus that is attacking your helper T cells. So the whole thing will enter, envelope and all, by endocytosis. And in order for the virus to uncoat, the viral envelope has to fuse with the endosome. So remember, if a cell is entering another cell or something's entering another cell via endocytosis, that means that part of the target cell membrane is really enveloping what's entering entirely. So you have this outer membrane that's part of the cell membrane forming that vesicle that thing just entered into. We need to be able to release, I didn't say we, the virus wants to release its contents into the cell. So you need the viral envelope to be able to fuse with the endosome that just took in the virus. That is the job of GP41. If this protein does not work, the virus cannot uncoat. In this case, it's uncoating, the capsid is broken open, and now the viral RNA is released into the cell. It does reverse transcriptase and makes double-stranded DNA. The DNA gets into the nucleus of the helper T cell and it inserts itself into that helper T cell DNA. You need a viral protein or enzyme in this case to do that, called integrase. If this doesn't work, if the integrase doesn't work, then the viral DNA won't be able to insert into your cells, and that will affect the overall infection of the virus itself. All right, so when it's in your DNA, then every time this cell needs to do transcription of something else, it's going to transcribe this piece of DNA and make more RNA from the virus, particularly messenger RNA, which is positive, so we know that that's going to be the actual genome of the virus. And now that cell can make viral proteins, including more GP120 and GP41. So the viral RNA, the viral proteins find each other, kind of collect, but don't assemble yet into a virus. Everything gets released, taking envelope with it, and then through the function of a viral protein called protease, an enzyme, then assembly occurs. So again, if you can mutate this protein, you can prevent assembly of those viruses and therefore prevent further infection by that virus. So this is the overall replication cycle, and again, I mentioned that there are a couple other web links that go over this in more detail. This next picture shows you specifically the roles of GP120 and GP41 in attachment and entry into the cell. So again, they're just showing you more detail. Here's GP120 attached to this CD4 receptor on this target cell. This could be a helper T cell. That's going to trigger endocytosis of the whole thing, including the envelope. Here's the envelope. Here's the endosome from the target cell. These two things have to fuse to one another to allow the virus to uncoat, and that is the function of GP41. So again, if you can target, if you're thinking about drugs to treat this virus, target either of these proteins and you're going to prevent infection. The problem is they change a lot and by mutation, so if you come up with one drug one year, it might not work next year. Okay, let's see. Targets. So I mentioned earlier, I kept saying helper T cells. There are other immune cells that have that same CD4 receptor on their surface, and that's what HIV is attaching to. Helper T cells are a huge target, but other cells like macrophages, dendritic cells, muscle cells, maybe liver, nerve, and epithelial cells, some of these are not even immune system cells, they also have that same receptor as well. So there is possibility that HIV can target more than one cell. I would say more often than not, it's targeting a helper T cell. And so think about that. You learned about adaptive immune responses, the cell-mediated response, and the antibody-mediated response. This is a virus, right? And so that technically should trigger the cell-mediated response. But even if it did trigger the antibody-mediated response, the fact that this virus is infecting helper T cells, those are the very cells that you need to do the cell-mediated response or to do the antibody-mediated response. So while your immune response upon initial infection of HIV can work, and one of the web links talks a little bit more about this that I posted, but you can have a successful immune response against HIV at first. But the problem is, if you cannot completely stop the spread of that virus, it's going to keep going to more and more helper T cells. And those are the very cells that you need to elicit the immune response. So the longer this virus can infect cells, the fewer helper T cells, functional ones, you're going to have, and the lower your immune response is going to be. So this is why HIV leads to the immune deficiency disease known as AIDS, because it wipes out your entire immune system. Not only is it wiping out the cell-mediated response, but because it's particularly targeting helper T cells, we know that those also help in the antibody-mediated response too. So it's wiping out your entire adaptive immune response, which in and of itself, the virus may not cause direct death, but the problem is you have no immune system now to fight off any disease. And so often in patients that have full-blown AIDS, they're dying from diseases that a healthy immune system would normally be able to fight off on its own, or maybe with a little bit of antibiotics or something like that, like pneumonia or something like that, which seems harmless to an extent to a healthy person, but somebody with full-blown AIDS, no, because they have no immune system to fight it, and no amount of antibiotics is going to help. So it's a kind of a clever virus. You know, if you're a virus and you want to survive in the world, and you infect humans, then it makes sense to infect a key player in the immune system, because you're debilitating that person's ability to fight it off. Let's see. This talks about kind of how it showed up in the United States. So this happened very early on in the 80s, and at one point they thought that the CDC thought that they knew the exact person who brought it to the United States. They named that person publicly. It was terrible for that person. And then it turned out that they were wrong. So I don't know for sure if they know who actually brought it over. But it did start in the male homosexual population in the United States, particularly in the San Francisco area. And so at that time, before there was a lot known about the disease, there was definitely a stigma associated with it. Now we know that anybody can get it. It is sexually transmitted and through blood. So through, if you're sharing needles, and if you're getting a blood transfusion and the blood has been affected with HIV, this happened to a lot of people in the 80s before they knew what it was. That was a way to get it. So I'm thinking about right now, a lot of you are too young to know what it was like during this time. Even I'm a little too young to really know what it was like, but I was old enough in the late 80s when it was still a problem, and there were still a lot of unknowns. And I'm comparing that time to our time right now with COVID-19. And it was really scary for a lot of people because they didn't know what it was. They didn't know how it was spread. They didn't know what was causing it. And everything that the CDC is doing right now to figure out COVID-19, they were doing with HIV. Working nonstop, trying to find out what's causing it so we can figure out how to prevent it. And now, let me go back. Back then, HIV, if you had HIV, there was a death sentence. And now it's not. It's very treatable now, at a cost, I have to say, but it is treatable. It is expensive to treat, but you can live with HIV now and not develop AIDS. So this was not that long ago. And so a tremendous amount of advancement in the technology behind treatment and just the knowledge of the virus itself has happened in not a long period of time. So if you're feeling a little worried or a lot worried about the current pandemic, it will get figured out. I don't know when, but it will get figured out. And at least now we seem to know how it's spread, which is a lot more contagious than HIV is, which in some respects makes that a little more scary. But we know how it's spread, so we know at least a little bit about how to prevent it. And as long as you follow the CDC guidelines, you should be okay. So in terms of HIV, I said it's sexually transmitted. It is found in your blood. It's found in your semen. Those are the two bodily fluids where the numbers of virus are high enough to cause infection, versus you might find it in saliva and breast milk, but it's not infective in those fluids. It's blood and semen. So sexually transmitted, and if, in a faulty blood transfusion or sharing of needles or things like that. Let's see. So how do you diagnose it? How do you diagnose AIDS? So HIV, there are tests, antibody tests and other tests you can do to see if you have virus in your blood. And they can even, there are tests out there right now that can detect how much virus you have in your blood. And so the goal of a lot of these newer treatments, especially if you've seen them advertised on TV, is to get the numbers of HIV particles low enough that you can't spread it to other people, and probably isn't spreading very quickly even in the patient. That is the goal. There's no cure for it even right now. There's no vaccine for it either, for those reasons I talked about earlier. But there are a lot of treatments out there that are pretty effective in keeping HIV levels low enough that a person does not even develop AIDS. If you were to develop AIDS, how is it diagnosed? Do you have HIV in your blood? Do you have low levels of CD4, meaning helper T cells? And do you have those other opportunistic infections? Maybe not all of them, but do you have a few? If those are all true, that's when somebody is diagnosed with AIDS. There are even some people without major treatment that never develop AIDS, even if they're HIV positive. What could the reasons for that be? They could just have really robust immune systems. People are still trying to figure that out to see, well, maybe we can come up with treatments from those people's blood that could be helpful in treating other people. In terms of treatment, it's commonly called ART, which stands for antiretroviral therapy. Usually it's a cocktail or a mixture of a variety of different drugs. The goal is to reduce viral replication. So going back to let me go back to that viral replication image. What could we target? If you can prevent attachment, prevent this fusion forming here of the endosome and the envelope, prevent insertion of viral DNA into the target cell chromosome, prevent assembly. Any of those things are going to help lower the replication of that virus, and therefore lower how much virus can be passed to other cells and passed to other people. So again, there's no cure. We're just trying to control HIV replication to keep the viral levels low so you can still have a functional immune system.